Hello everyone, so today we are going to start our chapter Genetics, lesson number 4 from biology book. That's the name in the book is Principles of Inheritance and Variation. So first of all, we will start with the definitions, some definitions of this chapter. So first we are going to study about what is genetics. Genetics is branch of biology in which we study about in which we study about inheritance and variations in which we study about inheritance and variations. Okay, that's called genetics. Branch of biology in which we study about inheritance and variations, that's called genetics. Okay, now we got 2 points here, inheritance and variations. So we will study about this. What is inheritance or heredity? Inheritance or heredity is transfer of traits, transfer or passing of genetic traits from one generation to another, that's called inheritance. Okay, and what is variations will be difference between parents and their offspring, yeah, children. Okay, what is variation? That difference between parent and their children, that's called variation. Now we are reading about DNA. The full form of DNA will be deoxyribonucleic acid. Deoxyribonucleic acid. Okay. And what is DNA? DNA is genetic material. DNA is genetic material in almost all livings. DNA functions as genetic material. Okay, and it's made up of polynucleotides. DNA made up of polynucleotides. Okay, next, RNA full form ribonucleic acid. And RNA do not function as genetic material, but in some viruses RNA functions as genetic material. Okay, in some viruses RNA functions as genetic material. Now what is chromosome? Chromosome is bigger part of DNA, and it present in condensed form. Now what is condensed form? It will be present at like this. Okay, it will be fully condensed or coiled. Okay, so chromosome will be like this. Now what is gene? Gene will be a smaller part of DNA. Gene is also a smaller part of DNA which represent itself and makes protein. So what is gene? Gene is a smaller part of DNA which represent itself and makes a protein. So this will be definition of gene. Next point will be allele. So what is allele? So allele is alternative forms of a gene. That's called allele. Now allele are of two types, dominant or recessive. Okay, so what is dominant and what is recessive? Dominant is the allele that present itself in every condition and also in the presence of recessive allele. Okay, so dominant allele will be it present itself in every condition and also in the presence of recessive allele. But what is recessive allele? Recessive allele can't represent itself when the dominant allele is present. So dominant present itself in every condition and also in homozygous and heterozygous. But recessive allele always present itself in homozygous condition. So we got here two words. What is homozygous and heterozygous? So homozygous what? Same types of allele. Same types of allele is called homozygous allele and different different types of allele is called heterozygous allele. Like we are going to study examples. Examples. Capital T. Both are same, so it will be homozygous. Small t. These are also homozygous. Capital R, small r, capital A. These all are the examples of homozygous alleles. Okay. So, what is a heterozygous allele? When the capital T and small t both are different, it's called heterozygous alleles. Okay. Capital R, small r, capital A, small a, etc. So we got here what is homozygous and what is heterozygous alleles. So now we are going to study about phenotypes and genotypes. Phenotype is observable physical condition. Observable physical conditions. Okay, of an organism. Of an organism. Okay, that's called phenotype. Like its height, color, etc. But what is genotype? Genotype is composition of genes or alleles of an organism. That's called genotype. And genotypes will always be written in homozygous and heterozygous condition. I repeat, genotypes will always be written in homozygous and heterozygous condition, or that's also called pure and impure. Homozygous like same, so it's called pure allele, and it's called impure allele, but these are not same, or heterozygous, so it will be impure या hybrid allele. Okay. So next our topic is Gregor John Mendel. So who was Mendel? About Mendel, we studied in 10th class also. So Mendel's full name was Gregor John Mendel, and Mendel first demonstrate about that transmission of characters from one generation to another, who was Mendel. So that's why Mendel called father of genetics. Mendel called father of genetics. Now Mendel worked upon garden pea. Mendel chose which plant? Garden pea, and he worked upon garden pea from 1856 to 1863. This is very important. 1856 to 1863. So, if we study about Mendel, so Mendel was born in Brunn city of Austria. Brunn city of Austria, and he was a father in church. Okay, also a mathematician or a botanist. Okay, now pea plant, that garden pea, that have seven contrasting characters. Contrasting characters means that have also their opposite characters, like stem height. If we take an example of stem height have two characters. Dominant character is tall and recessive character is dwarf. Okay, so dominant is tall and recessive character is dwarf. Next, shape of seed, that is dominant is round and recessive is wrinkled, like this is dominant round and this is wrinkled condition. Okay, next, color of seed. So dominant color is yellow and recessive color is green. And if we talk about pod color, like a pod of a plant that have color green and yellow, we know that green vegetable green vegetables and their pods have greenish in color, and and when they became mature, they turned into yellow color. But in the seed color, dominant color is yellow and the recessive color is green. Next, pod shape of pod, that in the dominant condition, when it will be greenish, it will be fresh, it will be inflated, and when it will be mature and old, it will be constricted. That's a recessive character. Next, color of a flower. Flower's dominant color is violet and recessive color is white. And the last one is flower position. Position of a flower. When the flower is present upon the top of a plant, that's called axial condition. And when the flower is present at the side or not at the top, that's called terminal condition. Next, our topic is, why did Mendel choose a pea plant? So reason for selection of pea plants are, first, pea plant is a bisexual plant, so that we can easily do self and cross pollination. Self and cross pollination, because it's a bisexual plant. Okay. Next point, it's a annual plant. That's why it can easily grown in every season. Okay, that's also beneficial. Next, small in size, so no need of a wider area, it can easily grown in a smaller area. Okay. Next, pea plant has a short lifespan of 3 to 4 months. Okay. Next, result a number of seeds produced many in number. Okay, so we got many number of seeds during this period. So we got many results. Next, it have limited characters or chromosome. How many characters? We already studied that 7 contrasting characters. Okay. Next, 3 other scientists rediscovered the laws of Mendel's and studied about that results of Mendel's. That was, we can heading this rediscovery of Mendel. Rediscovery of Mendelism. Okay, a rediscovery of Mendel. That 3 scientists was Hugo de Vries. Hugo de Vries. Second is Carl Correns, and third one is Eric von Schermark. Eric von Schermark. So these were the 3 scientists, Hugo de Vries, Carl Correns, and Eric von Schermark, that rediscovered about Mendel's results. Thanks for watching. We will meet in next video. Okay, bye.